Yeah. So Smriti, you can start the recording.
Yes, sir, I have started.
Great. Yeah. So all right. Is my slide visible? Is my Yeah. Okay. Great. Yeah. So last time we were discussing about the cyclodextrin, and I know which happens to be a polymer of glucose, where the glycosidic bond is alpha 1,6 linkage. Okay, sorry, alpha 1,4 linkage, and we discussed that because of the alpha linkage, between two glucose monomers there is a some sort of a bend. So it's more like a step, right? And because of the presence of a step kind of a pattern, uh, you know multiple monomers, when they join together, it leads to the formation of a bend or a kink, which is visible here in the it's my laser pointer. Yeah, so which is sort of visible in the amylose, right? And it turns out that if you just have a 6 or 7 of these monomers joined together, the head and the tail can come close enough so that there is a reaction between the first and the seventh or the sixth unit, and it forms cyclization, and that leads to the formation of cyclodextrin. And cyclodextrin, as we discussed, that it has great usage in terms of as a delivery vehicle, where you can encapsulate very hydrophobic molecules like taxol, paclitaxel, whatever, and deliver them into the blood vessels, and there is a spontaneous slow release of this molecule, and reasonable concentration can be achieved in the plasma circulation, as opposed to if you just use the drug, because the drug being very hydrophobic, it just tends to you know precipitate or accumulate at just precisely the site of action. Okay. All right. So just like cyclodextrin or amylose, glucose also forms polymers which are called as glycogen. I mean, probably most of you are aware of glycogen. Those of you are not aware of glycogen, it's a storage form of glucose, right? And glycogen is nothing but you have two different things in glycogen. So you definitely have the so yeah, so let's take a look. How do we? Yeah, so you have the C14 linkage, meaning that alpha 1,4 linkage, where the C1 is actually connected to another glucose through the alpha glycosidic bond, right? Okay, and this pattern actually continues, and it turns out that in some cases you also have the C16 linkage. Okay, so I mean, so that leads to a little bit of branching in glycogen. Okay, so you have for example, this building block, right? So in this one, what happens is the C6OH, which is sort of free in this unit, right? The C6 hydroxy group also engages in the formation of a glycosidic bond with the C1 of another monomer, right? So for example, if you look at this guy, right? So this guy, is engaged in both the C1 alpha 1,6 linkage, okay, with the C6 carbon atom of the C6 hydroxy group of another glucose. Likewise, it's also with the C4 hydroxy group, it's engaged in another glycosidic bond with another glucose unit. Okay, so that leads to some sort of a so imagine, for example, in this amylose, this guy is going in a very nice helical manner, but suddenly you have C6 hydroxy. So for example, this would be the C6, right? And another glucose unit can link here, and then you can have another branching through C1 C4 linkage. Okay, so linear polymer, along with that some branches. Okay, that is what you have in glycogen. All right, now yeah, so as you have the C14 linkage, which is an alpha linkage, just like amylose, glycogen is also sort of, bent molecule, which is sort of a helical molecule. It's soft in nature, and that can be shown by experimental means. However, there are other polymers of glucose. One of the most abundant polymer that is present on Earth, that would be cellulose, right? And you know, typically cellulose in it's more in the animal kingdom, plant kingdom rather than the animal kingdom. Now, cellulose is also a polymer of glucose, but it turns out that in cellulose, instead of a alpha 1,4 linkage, alpha 1,4 linkage would be like the C1 is of an axial, right? And the C4 is equatorial anyway. So C1, C4 would be here, right? That is what is found in glycogen and amylose and cyclodextrin. Okay, whereas in cellulose, what you have is a beta linkage, right? The equatorial hydroxy group, I mean, in the beta anomer, that engages in a 1,4 linkage, all right, with the next glucose unit. And that pattern essentially repeats, and because of which what happens is, uh, that so if you consider a single chain of glucose which is linked through the beta 1,4 linkage, right? And then, so it's not the polymer is not a bent molecule, okay? Rather, it's more like a linear molecule, because you not only have the C1 C4 which is connected to the covalent bond through ethereal linkage or glycosidic linkage. The here this would be considered, so the oxygen here, okay, which is the hemiacetal form. So this oxygen also engages in some sort of a hydrogen bond with the this would be 1 2 3 C3OH, right? And this hydrogen bond sort of stabilizes, the conformation of this single linear polymer, okay? And interestingly, what happens if you have multiple of these chains coming together, they can arrange in some sort of sheets, okay? So for example, you can see that, this single one, this is more like a linear. Imagine that, this guy which is shown from left to right here is shown from, let's say, from bottom to top or top to bottom, whatever. This is one long chain, and you have another long chain, you have another long chain, another long chain. They're all arranged in a parallel manner. And it turns out that, the other long chains, of course, you're not forming one linear chain, multiple linear chains are formed at the same time. So, similarly, you have other layer of these parallel monomers, parallel polymers. And these parallel polymers, you can imagine it's formed in the form of a sheet. Okay, this is one plane and this is another plane, and these two sheets, thin layer, can actually, they are sort of interlinked with each other. Interlinked meaning that they are arranged in a very nice stack, because and that is stabilized by the formation of intermolecular hydrogen bonds. So for example, you see this one linear form, okay, one linear chain is attached, non-covalently attached to the another linear chain via the formation of hydrogen bonds here, okay? So here, for example, this guy, this is an intermolecular hydrogen bond, and you have some sort of a intramolecular hydrogen bonds like this one here, okay? So in a specific plane, right? So in one plane, multiple not polypeptide but polymers of glucose can are stabilized by the formation of intermolecular hydrogen bonds, and one plane, in a one specific plane where you have parallelly occurring monomers which are stabilized by the formation of intermolecular hydrogen bonds, can also interact with another plane of this parallelly occurring monomers of not monomers but polymers of this saccharide, and those are also stabilized by this intermolecule. You can see this, this hydrogen. Well, okay, now this is for the sake of clarity, the plane has been taken above, but of course, these are not drawn to the scale. So it doesn't mean that, these hydrogen bonds are longer, because if you are to imagine these are long hydrogen bonds, there won't be any hydrogen bonds, because about the hydrogen bonds, you have a specific distance and geometry associated with the formation of a hydrogen bonds, right? And that is why, so you have multiple of these sheets sort of, arranged together, and that gives cellulose a very, very strong, you know character, right? They are very hard molecules like wood, for example, as opposed to glycogen or amylose, okay, which are much softer nature. And this softer nature of amylose or glycogen is associated with this. See, just a very simple thing, right? For example, if you just consider amylose, these amylose do not essentially associate with another chain of amylose, because the molecule is essentially bent, and since the molecule is bent, another chain of amylose cannot really come close to each other and form intermolecular hydrogen bonds and stabilize these chains. Whereas if you just have just a simple swap in the stereochemistry, one alpha to one beta linkage, and the molecule arranged in a very parallel manner, and many of these, this is beautiful cooperativity that you will find in nature, right? And you might also appreciate that how a single, just the presence of multiple of such hydrogen bonds, right, lead to the formation. See, essentially these are all single chains, right? These are all single chains, but all these single chains are essentially held together each other by the formation of intermolecular hydrogen bonds, right? And intermolecular hydrogen bonds can occur in the plane, across the plane, in different planes, and that essentially gives this molecule very, very hard nature, right? So this is what is essentially called as sort of cooperativity, meaning that many of these hydrogen bonds come together and give the molecule its very hard property, as opposed to glycogen, right, or amylose or cyclodextrin. All right. So okay, I will discuss this later. We'll come to something else. Okay, now like amylose, okay, just no, sorry, just like collagen, not collagen. What am I saying? Cellulose, like cellulose. There are also other polymers of glucose. For example, I'll just show you the molecule first. So this molecule is where is that? Yeah, here. So this molecule is called as chitin. Okay. Now chitin is also, here what you have is you have one N-acetyl glucosamine connected with another N-acetyl glucosamine. Okay. And they are linked through the beta 1,4 linkage. Okay. But the only difference between these molecule, this disaccharide and the disaccharide that we saw in cellulose, is the difference is the presence of a N-acetyl group, right? As opposed to the glucose, where you have a hydroxy group here, okay, here and here. And so that certainly, differs in the overall property or the overall hydrogen bonding pattern of this molecule. But nevertheless, since there is a beta 1,4 linkage, okay, so they can all arrange in the form of a stack or sheet, as we saw, and they can give this molecule chitin also very, very hard structure. Now chitin is something that is found in the exoskeleton of insects. For example, here this is a cicada, okay, and this guy is actually shedding its exoskeleton here. So here is the exoskeleton. It, of course, pretty much resembles the cicada, but this exoskeleton, it's composed of mainly chitin, but many times, this chitin is present in the matrix of I think calcium and other minerals, that gives this molecule even harder nature. Okay, so yes, this was about chitin, and there are other molecules. So for example, eventually in your biology lessons or not, in the practicals, probably you would be using agarose gel electrophoresis. Agarose gel electrophoresis is something that is used to purify a DNA. And in DNA, you would have agarose. And this agarose is also a very soft material, which is composed of galactose and anhydrogalactose, right? So here also you can see that galactose and anhydrogalactose are linked by a beta linkage. So this is a beta 1,4 linkage. and here the anhydrogalactose and the galactose is linked by a alpha 1,4 linkage, alpha 1,3 linkage. Okay, and but this molecule is, again, although you have beta and alpha linkage, that of course leads to a softer nature of this material. Moreover, this anhydrogalactose, you have this CH2OH, CH2O, which is in a form of a cyclic nature. And this molecule, unlike your cellulose, you don't have the free hydroxy group, which engages in a sheet across the plane intermolecular hydrogen bond. Okay, so likewise, okay, so this is about several polymers of glucose or monosaccharides, and how their properties vary just purely because of the certain stereochemistry, and those stereochemistry is essentially associated with the anatomy of this molecule, where there are occurrence of multiple hydrogen bonds. Sometimes you can block those hydrogen bonds, and that eventually reflects in the overall physicochemical properties of these molecules and so on. All right. Now, uh, with this, we'll just go to a very now in biological systems. So polysaccharides are not just present in the form of polysaccharides; they're also present as some sort of a chimera. Chimera in the sense that these polysaccharides are also sometimes attached to lipid molecules. Sometimes they are attached to proteins. Now, when they're attached to proteins, they have very important functions to play. For example, it's known that proteins undergo, two types of glycosylation. Okay, meaning that, protein chains, what are protein chains? We will begin proteins in this class itself. But before that, just want to finish the saccharides. And it turns out that, proteins, there are three specific amino acids on proteins which can get modified by carbohydrates. Carbohydrates meaning they're glycans. Okay, so for example, there is one amino acid. There is an amino acid which is called as a serine. Okay, so this serine, so this is the backbone of a. So here you see C double bond O. This is called as a C alpha. You have an NH group. Okay, and that's how the polypeptide chain essentially goes. And you have orthogonal side chains, meaning that side chains which are coming out of the backbone of the plane of the polypeptide backbone. Those are called as orthogonal side chains, and that determines the property of the proteins and the individual amino acids. Now, so here serine is an amino acid where there is a free hydroxyl group in the side chain. Okay. Now, because of the presence of a free hydroxyl group, so you can imagine that when you form a glycosidic bond, the hemiacetal form, so for example, the anomeric center engages in the formation of a glycosidic bond with another sugar molecule where there is also another hydroxy group, right? For example, you have C3 or C4 hydroxy groups. They engage in the formation of a glycosidic bond. The same thing here. So serine, now serine is an amino acid where there is a hydroxy group in the side chain. There is another amino acid which is called as a threonine. Okay, that also has a hydroxy group, but there is some little difference in serine and threonine. But these two amino acids undergo glycosylation. Okay, so where the glycosyl group, okay, or the glycan chain, or a single monosaccharide essentially is linked to the oxygen of these amino acids. Now, this is called as a O-linked glycosylation. Okay, O-linked, meaning that the oxygen from the amino acid engages in the formation of a glycosyl bond with another monosaccharide. Okay, you have something which is also called as N-linked glycosylation. Now, what is N-linked glycosylation? N-linked glycosylation happens on, so you can the name intuitively suggests that it's associated with nitrogen, right? So there is an amino acid which is called as asparagine. Okay, so in asparagine, this is the polypeptide backbone, and the orthogonal side chain has a CH2 CONH2 group. Okay, This NH2 group, which is also a amide, okay, CONH2. Okay, now the CONH2 can get modified by the formation of another glycosidic bond, okay, and this is called as a N-linked glycosylation. Okay, so you have two types of glycosylation, N-linked glycosylation and O-linked glycosylation. Okay. Now, O-linked glycosylation is associated with serine and threonine residues on the protein backbone, and N-linked glycosylation is associated with asparagine side chain. All right. Now, but it's not that they differ only in the chemistry of the glycosyl attachment. They also differ at the sites where this N-linked and O-linked glycosylation takes place. Okay, so it's known that N-linked glycosylation is which is actually which is the number of N-linked glycosylation is a lot more. The N-linked glycosylation is a lot more prevalent than the O-linked glycosylation. Okay, and it's known that the N-linked glycosylation is associated is more like a co-translational process, meaning that as the protein is being translated or I mean or let's say being it's synthesized, okay, then the glycosylation spontaneously takes place on that. So for example, this is something which is called as endoplasmic reticulum. So don't worry about the name, it's more of a compartment where the protein is synthesized here, and as the protein is being synthesized, okay, a part of this essentially sticks out, okay, where there is the specific recognition sequence. Okay, now that will also tell you in a minute. So there is a specific site. For example, here you can see there is an asparagine which is shown in a yellow Y kind of stick. Okay, and this asparagine which undergoes an N-linked glycosylation, that is let's say it's templated by an enzyme which is called saccharide transferase. These are class of enzymes which are called as oligosaccharide transferase. Now, so what happens, and you also have molecules, for example, here. So here you have a 14 sugar unit containing lipid. Okay, this lipid is called as a dolicol. Okay, now this is actually again hanging onto the surface of the endoplasmic reticulum. Now, so what this oligosaccharide transferase enzyme does, it sort of brings both of these guys together, okay, and bringing both of these two guys together, because there is a specific pocket, there are a specific let's say it acts as a dye, right? It dyes. Okay, so because so there is a specific site for the asparagine to dock, and there is also specific site for this dolicol containing mannose whatever unit to dock, and then this guy essentially transfers this entire 14 residue uh, polysaccharide unit onto the asparagine chain. Okay, and that's how the entire oligosaccharide is transferred onto the polypeptide chain. Okay, so this is what is so N-linked glycosylation, as I said, that it's a co-translation process. Okay, where the entire large not polypeptide, but the entire large oligosaccharide unit, and that is why this guy is called as a not a glycosyl transferase but an oligosaccharide transferase. This transfers the entire unit onto the asparagine. Okay, now okay, whereas I don't know whether I have this. No, I don't have it. But just to mention that, for example, and as opposed to N-linked glycosylation, the O-linked glycosylation are that's not a co-translational process. Okay, so O-linked glycosylation are which are taking place onto the serine and the threonine residues. The O-linked glycosylation and there are glycosyl transferases which do this job, meaning that it's not a co-translational process. Once the protein is essentially synthesized, then the O-linked glycosylation takes place. And the main difference between the O-linked and the N-linked glycosylation is, whereas in N-linked glycosylation you have large oligosaccharide units which are linked onto the asparagine, in the O-linked, on the other hand, one unit at a time is essentially being linked. Okay, so here you can see this is the O-linked glycosylation. Here you have a GalNAc residue which is attached, Probably if there are enzymes present, there may be one or two more units of this, either galnac or glucose or galactose, whatever will be attached, or mannose. Okay, so it's not a large oligosaccharide unit like 14, 15 mers. Okay, so that is the fundamental difference between the N-linked and the O-linked, and of course the site of modification is also different. All right. Now the other thing, why, now the question is, for example, why do what is the reason, or what do we know about that that glycosylation of proteins? What sort of functions do glycosylation of proteins play, right? So one very important thing that people have realized over years, for example, I think this protein is an erythropoietin. Okay, so this erythropoietin is sort of associated with let's say formation of you know red blood cells. Okay, now it turns out that erythropoietin, okay, erythropoietin essentially circulates in the blood. Now it turns out that the if more of the erythropoietin is circulating in the blood, the more of erythrogenesis. I'm not sure whether erythrogenesis is the right term, but so more of red blood cells is essentially produced by the liver. Okay, so now the question is why. Okay, and it is also known that the number of glycosylation. Okay, so this is N-linked glycosylation. The more glycosylation that this erythropoietin has, the more erythrogenesis. Well, the more is the, let's say that the more is the activity of the protein. And the question is why. It turns out that so just take a look into this guy. Okay, so it turns out that as we read or know about the proteins, you will see that proteins are composed of polypeptide bonds, right? Now polypeptide bond is nothing but it's a CONH linkage. Okay, it's an amide linkage. Now there are now amide linkages, there are many enzymes in our blood, or in multiple places, and even in our gut, everywhere essentially. Okay, everywhere in our body, which are responsible for cleaving these proteins. Okay, and the way they cleave these proteins, I mean, and these enzymes are called as proteases. Okay, so and what these guys do is they essentially cleave the CONH bond. Okay, and they cleave this, and by cleavage of these polypeptides or proteins, what you have is you have the complete loss of the structure of this polypeptide or the protein. And as you will study probably later, that the structure of these proteins are intricately linked with their function. Okay, because the structures, so these protein structures are essentially sort of scaffolds which hold the specific orthogonal amino acid side chains in a particular orientation, in which they can essentially go and dock onto the surface of their target. And when they dock onto the surface of the target, the role is played. Okay, so whatever, if there is conformational changes, if there is translocation, whatever. Okay, right. Now now what happens is, if you, for example, if this molecule, imagine that you are to break this bond. Okay, so if you are, so this is, so these sheets, these arrows essentially show a specific conformation of a polypeptide chain. Okay, and this conformation is called as a beta strand. Okay, you will see later what are beta strands, but but essentially this is a single polypeptide chain, CONH repetitive CONH, and in between you have the C alpha units. Now the CONH, if the CONH is essentially cleaved by a protease, what will happen is this arrow will break apart, right? And if this arrow breaks apart, what can happen is, if you have a hydrophobic core in the protein, that hydrophobic core will be exposed to solvents, and of course that will lead to total loss in the conformation or the three-dimensional structure of the protein. Okay, so how do we prevent it? Okay, if there are ways to prevent it, nature would have done it, right? And that's what nature does by glycosylation. Now what nature essentially does by glycosylation is, now glycosyl groups, for example, as you will see later, that to form a such a globular structure, you need many amino acids. Let's say that probably you need 500, or not 500, maybe around 150 amino acids, 150. Okay. However, you see that for example, if you have a very simple oligosaccharide unit, you probably can even count. There will be probably 13 or 14 oligosaccharide units, which essentially covers a large surface area on the protein. Okay. Now, what happens, by covering this large surface area of this protein, what this guy essentially does is it prevents the attack of the onto these polypeptide chains. Okay, so this oligosaccharide units acts as a barrier for the protease to act onto the polypeptide chains and cleave it. Okay, that's how these oligosaccharide units can prevent the proteolysis of polypeptides or proteins. Okay, and many times it is also observed, we are not going to deal with it here, and it is observed that oligosaccharide units can stabilize the folded state of the protein, because people have seen that, for example, okay, so you would remember, when we were discussing about the cyclodextrin, right? And we were saying that, the inner core of the cyclodextrin is pretty hydrophobic. Okay, and whereas because the hydroxy groups here, they actually point outside, okay, in the periphery, but in the core, so in the top and the bottom of this glucose or whatever mannose is sort of hydrophobic. And since this the top and the bottom face of this six-membered pyranose ring is pretty hydrophobic, what happens is, there are some amino acids, for example, hydrophobic amino acids. We won't take the names. Those hydrophobic amino acids are very nicely stabilized in the presence of these additional hydrophobic patch. Okay, and that is how. Then there are other interactions also. Something which is called as a CH pi interaction. I mean, if time permits, we will discuss that. Okay, or maybe I'll just quickly tell you what happens is, for example, you know that let's take a look at this. Yeah, for example, yeah. So here you see that this is something which is a very weak hydrogen bond. Now, what happens? So this is your pyranose ring, and in the pyranose ring, you can imagine there is a hydroxy group here. Okay, and since there is a hydroxy group, not only this OH bond is polarized, that's why this hydrogen engages in a hydrogen bond. This CO bond, because of the difference in the electronegativity, this CO bond is also polarized. Okay, same for every other site. Okay, and that is why what happens, the hydrogen that is present at this specific site or this site or this site or this site, all of them are more acidic. Okay, and since all these hydrogens are acidic, they are sort of acidic, meaning they're sort of electron deficient. Now, since that is an electron deficient center, or this hydrogen is electron deficient, which is the same in the formation of hydrogen bond, this H takes part in a strong hydrogen bond because this H is electron deficient because of the high electronegativity of the oxygen, right? And the same thing here, the hydrogen associated here, that is also electropositive. And since this is electropositive, if you have electron rich rings, that is, for example, you will see that there are amino acids. For example, you have phenylalanine, where you have a phenyl ring there, a benzene ring. You have tyrosine. Tyrosine is a benzene with a hydroxy group. You have tryptophan. Tryptophan is something where you have an indole group. Okay, these are all aromatic amino acids, meaning that you have a electron cloud which is entirely delocalized onto the surface, and those guys can come very close, in close proximity with this. And since, although this ring is not really planar, but when we studied the conformation, you saw that they prefer to be in a chair like conformation. Here you see that this is present, they are present in a chair like conformation. So for example, this hydrogen which is present in the axial position, this hydrogen can engage in some sort of a CH pi interaction with those aromatic amino acids, and that can actually stabilize proteins, okay, at specific sites. Okay, this is exactly what happens. These kind of non-covalent interactions are very well known in in proteins which are actually glycosylated, and that's how they can provide stability, additional stability to proteins by getting glycosylated. So by glycosylation, you can have for example, as we saw discussed that, so the proteolytic stability of proteins can be increased, the folded state stability of the proteins can be increased. Okay, so yeah, so this is what happens. And just one last thing about glycosylation is, for example, glycosyl groups are also recognized by antibodies. Okay, so for example, the blood group type that you have, that's OAB and the AB. So it turns out that, so in the blood groups, blood cells are developing erythrocytes and so on. So you have glycosylation on the surface of this RBC. Okay, so for example, so in RBC, in the O blood group type, you see that, for example, you have the fucose, galactose, N-acetylglucosamine, and N-acetylglucosamine, and other galactose unit. Okay, so it turns out that this combination, okay, so where you have the fucose, 2 galactose, and 1 N-acetylglucosamine, this guy is not really antigenic. Okay, meaning that it does not elicit immune response. There are no antibodies, that sort of recognizes this. Okay, so but if this core unit is essentially further modified, for example, if it's modified by the formation of another N-acetyl galactosamine, or in this case, then there are specific antibodies that recognizes it. Okay, meaning that, your immune system will know that, this is myself. Okay, uh, this is myself. Okay, let's just come to the next sentence in a few minutes. Okay, likewise, if you have the same core unit which is modified by another galactose, it will know that, this is, there are antibodies which will be raised against, this kind of pattern, and that will know that, okay, this is my own. Okay, and likewise, if you have two of these guys, right, this thing here, if you have two of these guys, and then this is more of an AB group. Okay, so here also, you have an antibody response. But then, what happens, that is why you probably would have known, I mean, you might know that, if you uh, uh, okay, so now, so imagine that, I have a blood group. Okay, so I have this. Now if I am transfused with a B, okay, so what happens is, since my system, when I was born, or I was in the fetus, my system already knew that this A pattern, okay, this pattern is my own. As soon as I am given blood from this pattern or this pattern, my body starts raising antibody, sensing that, this is foreign blood group. Okay, and this is foreign, not only foreign blood group, it's a foreign particle that I have to destroy. Okay, so that is why, if you have a blood group A which is essentially transfused to B, or AB, or vice versa, you start raising antibodies, and these antibodies can cause really severe response, causing lysis of red blood cells and even death. Okay, however, on the contrary, if this guy is essentially given O blood group, then of course it doesn't raise antibodies against it. Okay, because the system sort of I mean, it doesn't know that to raise antibody. It recognizes, okay, fine, this is myself. All right, it's the same for B. B is transfused with O, or AB is transfused with O, no problems. Okay, so then you can essentially transfuse any of these guys with O, and you'll be fine. All right, and this is all because of the antibody response that you have because of the glycosylation patterns. Okay, right. So this is all about glycans. Now, if you have any questions, you can ask me before we go to the protein part.
Hello, sir.
Yes.
If the blood is RH negative, then also the same story will happen.
Oh, this I have not. I'm not very confident. I have to read and tell you. Okay,
Okay,
I
sir.
am not very familiar with the ratio system. Yeah, positive, negative.
So why doesn't the amylopectin also fold onto itself like amylose?
I didn't get it. Once again, please.
So similar to amylose, why
Yeah.
doesn't the amylopectin also fold onto itself to form helix?
Why doesn't amylopectin? Uh, How do you know that amylopectin does not form a helix? Why do you say so?
Like we have studied it in 11th.
So what is the modification in amylopectin?
It is slightly branched.
And it's not helical. Is that what is said?
Yes.
So like from a chemistry perspective, amylopectin was said to have C1, C6 branches and it resembles cellulose to some extent.
No, C1, C6 branch would not resemble cellulose.
Not cellulose.
Resemble
That would be
Glycogen. It resembles
Yes.
glycogen somewhat.
Yes. So it will be bent. It will be bent. I mean, maybe your textbook was comparing with respect to probably cellulose. I have to look up. I have to look up original literature. Then only I can comment. Because just purely by if there is an alpha 1,6 linkage, the alpha 1,6 linkage will lead to its bent nature. I mean, there is no alpha 1,4 linkage. Okay.
So
Just
can
a very virtue. It's like a step manner. It will be. There is no way it won't be, because the steric hindrances would not allow it to adopt conformation, which is like a straight chain that you observe in cellulose. Okay.
Sir.
Yeah.
Sir, you talked about blood transfusion. You said that when we transfuse O blood or the blood of O blood group into A, there wouldn't be formation of antibodies against O because there's no antigen on it when we are talking about A and B antigens. But sir, the blood group of O, that the blood that we transfused O blood group, that already has antibodies against A. So there would be a reaction, right?
I did not understand. What do you mean
Sir,
antibodies?
can you get to that slide?
Yeah. Here.
Yes, sir. So I'm saying that like when we are like the A1, it's O blood group, right? And we are transfusing it into the body of a person who has A blood group. Okay.
Right.
So
Right.
when we do that, the person with A blood group, he wouldn't form antibodies against O because O doesn't have A or B antigens on it, right?
Right.
But the O blood group, it already has antibodies against the A antigen.
No, of course not.
The
Why? Hold on. Maybe you need to clear one thing. So why?
Okay.
For example, so imagine I have O blood group. Okay. Why
Okay.
would I have antibodies against A until and unless I am exposed to? See, antibodies is something. So the foreign particle is called as antigen, antibody generator. You can say that. Okay. So until and unless I am exposed with A blood group, my body will not raise antibody against A. Okay. So O blood group itself in the blood does not have anything which is against A.
Okay, so because so, yes, this is something that we learned. I'm not sure, but I think so. We learned in our school level that the O blood group, it has no antigens but has antibodies A and B.
I am not sure.
I guess
That's like
I
okay.
uh, those with O blood group have the capacity to produce both the antibodies, and if one of the antigens is present, then the other antibody cannot be produced.
But did you understand my rationale? What I said?
Yes, sir.
So that's how it works in a standard antibody antigen response.
No, sir. I am a different person than the person asking the question.
Yeah. Okay. All right. Anything else related to glycans?
Excuse me, sir.
Yes.
Sir, when we talked about the N-linked glycosylation, sir, there were 3 steps that you told.
Yes.
Like less this one.
Mhm.
Sir, is it the same with the O-linked also?
No.
Like you told, like
No.
like,
No, it's not. It's not. So it's not the same. So okay. So O-link glycosylation. If I am not wrong, I think it takes place in the Golgi bodies. Please check it. Okay, I may be wrong here. I think it takes place in the Golgi bodies, and this is in the endoplasmic reticulum. It's a different compartment. And the other thing that happens is, for example, the O-link glycosylation is I mean, of course, you got that point that you don't have large oligosaccharide units, you have monomers. So the monomers that are attached onto the specific sites in the O-link glycosylation, it all depends upon the transferase enzymes present at that specific time, at that specific compartment, also the concentration of the transferase unit and the concentration and the availability of the specific monosaccharides. Okay, did you follow what I said?
No, sir. I don't
You did
understand.
not follow. So,
Please repeat, sir.
okay, so the first difference is there is no lipid tethered oligosaccharide unit in O-link glycosylation. Okay, that is first difference.
Yes, sir. So, like you said, there will be a monomer.
There will be monomer, yeah. Okay, and once a monomer is attached, if there are other oligosaccharides that can not oligosaccharides, but if there are other transferases, okay, glycosyl transferases, that monomer can go into a dimer also. Okay.
Okay, sir.
Meaning that can be disaccharide instead of just a monosaccharide, but one step at a time, meaning first a monomer, then a dimer, then a trimer, so on and so forth. Okay.
Yes.
Whereas in this guy, so here what happens is this large oligosaccharide unit is in one step, it's transferred onto this asparagine. But in that case, what happens is there are different enzymes for these different steps. So for example, the first one can be glucose, the second one can be mannose, the third one can be fucose. And for each of these, you need different transferase enzymes. All right.
Yes, sir.
Okay, that's how it essentially differs. But one thing I forgot to tell you that it's not that any asparagine undergoes a glycosylation. There is a specific recognition sequence. For example, it's known that, if I'm not wrong, it's asparagine, X, and serine or threonine. So you have to have an asparagine and followed by any amino acid, and after that you have to have a serine or threonine. Then only then asparagine can get glycosylated. Okay. Otherwise,
Excuse me, sir.
Yes.
I have one confusion. So, like in this case, the when we have that dolly call there, its core is hydrophobic. So that's why if it cleaves some, like the part cleaves, so there it's like components will actually go somewhere, like it won't attract and it won't form that we want to. So that's why it's going glycosylation. Is it like this or like? Hello.
No, do you mean to say that it's a very non-specific recognition here at my pointer side? Is that
Like
what?
the reason of glycosylation is what? Like I'm confused in the reason of glycosylation.
Sir,
Like
is
why
it
is
like
glycosylation
bodyguard?
happening?
No, I didn't. I am not sure whether I clearly understand your question. So are
So
you
like
saying that it's a random process where these dolicol units come in proximity to this blue guy and then gets phosphorylated? Is that what you're saying?
I mean that enzyme that we have, that transferase enzyme.
Yes.
Like the dolicol want to attach with the asparagine.
No, the
But it can
dolicol is a lipid tail. The lipid tail doesn't get attached.
okay, like
See,
that
the whole point, hold on. The whole point is, see, when enzymatic reactions happen, you will see later that, any chemical reaction, as a matter of fact, any chemical reaction takes place when there is a collision, right? If there are no collisions between A and B, there won't be the formation of AB, right? So this is A and this is B. Now, to increase the efficiency of collision, what you are doing is, since in the endoplasmic reticulum, the protein is being synthesized and this comes out from this region. Okay. So the oligosaccharide also has to stay in proximity if it's freely floating in the cytosol. So this part, this white part is essentially cytosol. Okay. It's cytoplasm. So in the cytosol, if this guy is freely floating, the efficiency will be very low. Then it will be like that means that you have to have another enzyme which will essentially bring this guy in proximity to that. Here what happens is this large oligosaccharide unit is already tethered to a lipid in this membrane. Okay. And we have seen that you have motions of lipids in the membrane, that FRAP experiment, right? So there is collision, of course, and what happens is it just due to the tethering of this entire unit, because this entire unit cannot get embedded into the membrane because this is very, very hydrophilic because of all the hydroxy groups. Okay. So just to increase the possibility of reaction, this guy is tethered. Does it answer your question?
Yes, sir.
Maybe partly. Think about it.
Yes.
If you still have questions, you can write me a message. Okay.
Hello,
Anything?
sir.
Yeah.
Isn't it a kind of a bodyguard for the protein,
It is.
which prevents it from proteolysis?
It is.
Okay.
It is. Yes.
Sir,
Anything else?
sir, I didn't understand why cellulose does not form a helix. Can you please go to that slide again?
Yeah. Cellulose, right?
Yes, this one.
Now, cellulose. So just look at the disaccharide unit. Forget about this. Okay. So you have one unit and you have another unit. Okay. And this one unit and the another unit. So this is a equatorial OH, ideally, right? When it was present, if it's not involved in a glycosidic bond, right? This C1, since we are talking about here, the beta anomer is engaging in the formation of a glycosidic bond. So this C1 is also beta. Okay. So you have two beta guys coming close to each other. So it forms this kind of a linkage. But if it was a beta, because C4 is always, equatorial, and if this was alpha, okay, so you will have this guy not aligned very nicely with this guy. Instead, this guy will go a little bit down because of the very nature of the axial equatorial bond. Do you follow? So
Yes,
this
sir.
would be axial, then equatorial, axial, equatorial, axial, equatorial, axial, equatorial, axial, equatorial. That's how it proceeds. So you have a step kind of function. Whereas this is equatorial, 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 like this. That's how it proceeds. And that is why this is linear. And the movement of this guy is further restricted by the formation of the hydrogen bond. Well, you can say it intra because the same polymer, intramolecular hydrogen bond. Does it answer your question?
Yes, sir.
Okay, if there are any more questions, ask, or else we'll go to the amino acids.
Hello, sir.
Yes.
The step-like function converts into helix structure because of the rotation of the sigma bonds, right?
Uh, Rotation of the no. I mean, which rotation of the sigma bond? Hold on. Which rotation are you talking about?
When you say that it is a step-like structure, then
Yeah.
it is in a single plane.
Sorry.
When you say that there is a step-like function formation, a
Hmm.
step-like structure formation. Right now, just few moments back, you said that.
Yeah.
So it is a somewhat planar one. All the steps are in the same plane, but a helix is a 3D structure. So when the
Steps are in a same plane. Yeah. Right. But if
and
you
the helix
okay,
is a 3D
yeah, yeah,
structure.
fine, fine.
So
Hold on. And helix is a three-dimensional structure.
yes,
So is cyclohexane a planar structure?
Not in that sense, but the centers of all the glucose units are in the same plane.
Centers of all the glucose units are in the same plane.
In that sense.
Centers of all those could be fine, no problem.
And now when you are talking about the step like the axial equatorial, that steps are formed.
Yeah.
In that also the centers of all the glucose units are in the same plane. So to convert it into helical structure, there must be the rotation around about the
Oh,
glycosidic
yeah, there will
bonds.
be rotation. There are rotation, of course. Yeah, see, for example, in this, well, I mean, you don't take my, see, I just tried to explain in terms of the dimer formation, okay, disaccharide, as opposed, I mean, in cellulose and the amylose. All right. But now, if I am to consider multiple of these guys coming in a pattern, okay, which is what you are having here, right? So, well, yeah, it's not a helix. It's not a helix. I mean, it's well, how do I call it? It's more like a curved ribbon or something. I don't. It's not a helix.
Ah, yes, a ribbon-like structure.
Yeah, it's not a helix. You're right. I used the wrong term. It's not a helix. Okay. So here
Yeah.
it's more like a ribbon kind of a structure. Okay, where there is a curvature associated with it. But what you must also appreciate that this step kind of structure that I mentioned, the step kind of structure is, I mean, if you are to only consider the linkage, but there are associated steric interactions which I'm not talking about. Okay, and that will lead to certain rotations in the CC bonds, and that leads to the formation of this ribbon-like structure.
Okay, sir, I understood completely. Thank you, sir.
Anything else? I mean, I think it's almost 12. It's pointless to start about amino acids.
Excuse me, sir. So
Yeah.
you had mentioned that glycosylation occurs through three amino acids.
Yes.
Which is the third amino acid?
Threonine.
Okay.
Threonine.
Yes, sir. Okay, sir. Threonine. Thank you, sir.
Threonine also has a OH group.
Okay,
Right?
so it's similar to serine.
It's similar to serine, yes, but I'm not sure how prevalent is that serine because of steric reasons. But yeah, there is a possibility. I think it happens, yes.
Okay, thank you, sir.
Sir, sir, why doesn't tyrosine make a bond? Because tyrosine also has the OH, like it should.
Yeah, that's a good question. But see, essentially, if you think of the linkage per se, okay, the chemical nature of the linkage, that is a phenol, right?
Yes.
Right. So that is a phenol, and if you are to think in terms of the mechanism, right? So you have that OH group there, right? I mean, here serine, you have an aliphatic OH.
Yeah.
Okay. So this aliphatic OH, the way the reaction proceeds is this lone pair of electron undergoes some sort of a nucleophilic addition onto a electrophilic center, right?
Yes. Oh, sir, like you mean that in the phenol case, that like lone pair is delocalized, so like it can't react.
Yes, and that is why
Oh,
since it's delocalized, your phenolic H is, the pKa is much lower, right? Phenol is acidic. I'm sure that many of
yeah.
you would have used phenol in your, I don't know, class 12 practicals. We used, and phenol is pretty acidic. You can use it, take a litmus paper, and then check it out. Whereas alcohols are certainly check the pKa. That is the reason, isn't it? And
Yes, sir. Yes, sir. Yeah.
and that is why alcohols, for example, if you take acetic acid, it can very nicely form, you have ethyl acetate, sweety smell. But I'm not sure whether you have phenyl acetate or something. That's the simple reason. However, if you have a benzyl alcohol, right, Instead, so you have in an intervening CH2 between that oxygen and the phenol and the benzene, that will undergo esterification. That will behave the same way like this. Followed?
Yes, sir. Yes, sir.
Okay. Anything else?
Sir, but we have aspirin where oxygen is bonded with the alcohol group.
We have what? I didn't get your question once again.
So he said aspirin.
Aspirin.
No, sir. I think he meant asparagine.
Okay. Asparagine is what?
No, sir, I said like there is a medicine aspirin there in the phenol. Oxygen is bonded with the other groups. As you said that benzyl alcohol can bond, but in threonine it cannot bond.
Yeah. See, you can form things, but so depending upon the how harsh the conditions is, right? Don't. We are talking about the glycosylation here, and then the esterification, simple esterification. Even if you form, for example, if you form a benzyl, not benzyl, but let's say phenyl ester or something, where CH3C double bond O, you have OPH, that will be extremely unstable just because of the little presence of water. That's a very good living group, right? And it will just immediately break apart. Do you understand what I'm saying?
Yes, sir, got it.
You can of course form phenolic ethers are known. If I'm not wrong, there is a natural product called as vancomycin. In vancomycin you have those kind of linkages, but that's a ether link.
Sir,
Yeah.
may I add something to this?
Sure.
Yes, sir. So as far as I think the reason why aspirin works as a medicine is because that phenolic linkage is unstable, and
Fine.
the aspirin group actually is released and it makes changes to
Okay.
protein.
Okay, fine. I mean, good. Your friend answered your question. Thank you. Anything else? Am I still audible? Hello.
Yes.
Okay.
So,
Yeah. So if there are no more questions, I think we can end this class, and we can. I mean, in the next class, we'll talk about proteins and amino acids. Okay. All right. Thank you, guys. So Smriti, you can stop the recording. I'll load the. I'll send Smriti